Good evening, ladies and gentlemen, and welcome to this National Equine Forum webinar. I'm Tim Brigstocke, I'm Chairman of the National Equine Forum, and I'm delighted that you've all come to join us at this webinar of Just in Time, Using Science to Save Our Breeds. This is the first of our special interest webinars which precede the 29th National Equine Forum, which will be held on Thursday, the 4th of March, I'm afraid virtually, as you would expect, and we'll give you further announcements about what's happening as the months progress. Just before we start, a few housekeeping announcements. First of all, the webinar is being recorded. Secondly, we're hugely grateful for the support we continue to receive from all sorts of organisations. And for this particular webinar, the sponsors of this are the British Horse Society and the Horse Trust, and the corporate friends are Weatherbys Scientific and World Horse Welfare. And then the friends are 826 Equine Studbooks Association, the Central Prefix Register, Cob Care, Exmoor Pony Society, New Shule, James Rayner, Traditional Gypsy Cob Association. Just moving on to some general housekeeping, you can read it as well as I can, but there are links. Please post them in the chat box as we go through the live webinar. You can see the programme on the resources page, and the details are there on the National Equine Forum website. The speakers' bios are on the webinar page. The recording link will be circulated 48 hours after the live webinar, and there'll be access for 2 weeks to watch the webinar on demand. And it's very encouraging, the amount of people who said they can't join us tonight but would like to see this webinar. And then finally, and perhaps most importantly of the lot, to make sure this is as interactive as possible, if you do want to post questions, can you put them in the Q&A box, not in the chat box, in the Q&A function. So that's all from me. I'm delighted now to be able to hand over to Tim Morris, who will chair tonight's webinar. Tim, over to you.
My name is Tim Morris. I'm a member of the organising committee, and I'm also the Vice Chair of the Rare Breed Survival Trust, the national charity that has a primary interest in our native breeds of livestock, and especially for tonight, our native breeds of horses and ponies. You'll see in the background some wonderful Highland ponies. The bad news about the current health situation is we're having to do this by a webinar. I should have been where these horses are in Scotland today. However, it does mean that nationally and internationally, we can get a really wide range of participants attending, and it can be recorded, as Tim Brigstocke has just said. We've got 4 great speakers this evening, time for discussions. There'll be some clarification questions after each presentation, just 5 minutes, and then a panel discussion, which can then extend further. So getting down to the nitty gritty, we should remember, and we're going to talk about biodiversity, that 50% of genetic diversity in a species is in the breeds. So breeds are really important. We've heard a lot about biodiversity in the last few days. The Prince of Wales has been talking about it internationally, as well as our own politicians. And the UK is a signatory to a number of different international treaties, and that is monitored by work of the Joint Nature Conservation Committee. So what do they say about our livestock species biodiversity? Well, what they say is this graph here, which is available on the resources page, but you will see close to the bottom a dotted line, and then just above that, a blue line for horses. And this is a measure of what's called effective population, which accounts for the number of animals in a population and the relative numbers of sires and dams. And males are as important as females, not for political correctness, but as you'll hear from Andy Dell, that balance is vital to a healthy breed. Basically, a low effective population size signifies a greater likelihood of inbreeding and loss of genetic diversity. And if you go and look at this graph in detail, and as I said, you can see it in much greater detail from the resources page, you'll see that many equine breeds are perilously close to the area where there's a real concern of extinction, which you're going to hear about later. So the other way of looking at this is to consider rankings, and the charity of which I'm a trustee, the Rare Breed Survival Trust, you may be aware of its watch list, and all the British breeds are on the areas of concern. And then there is actually a government committee chaired by our very own chair, Tim Brigstocke, the Farm Animal Genetic Resources Committee. And this publishes a number of data graphs, and you can go and look at the great detail for any breed you're interested in. And at the bottom right is, for example, the change in effective population over time for the breed of interest. So there's a lot of data out there, but as we'll hear later, I wonder whether we're using it to its good effect. So the speakers tonight: Tullis Matson. Many of you all know him and his family for their work with horses over several generations. But he's actually going to start with a wider view of endangered wild species, which we might say we all know about, but then draw it back to horses and perhaps give us a wake-up call when he starts talking about concepts such as extinction vortexes and how they might apply to the breeds that we are perhaps too comfortable about. Then we come to Simon Cooper, co-chair of the International Studbook Committee for Thoroughbreds. Of course, this is a UK native breed, although it's distributed around the world. And actually, there are some really interesting preliminary findings which shows that concerns about genetic diversity are even affecting such a large breed, large in size breed. Then on to Paul Flynn from Weatherbys Scientific, based in Ireland. A major part of their business, you may know about Weatherbys in terms of thoroughbreds, but they also run a very large cattle genomics business, but actually are doing more and more with the equine sector, especially since the sad demise of the Animal Health Trust. He will describe how genetic and genomic science can help native breeds of horses, with a very useful example from the local Kerry bog pony. And then last but certainly not least is Andy Dell, who's done so much with flagship work on the Cleveland Blay about managing breeds and breed society policies using genetic management. So the overall message here tonight is that there is a serious concern, but there are things we can do about it. We'll hear these presentations, we'll take some specific questions, and then finally, we will open up a panel discussion. Thank you for those who already submitted questions, and I'm sure there are going to be more.
Firstly, just to really thank the National Equine Forum for inviting me to do this presentation today. And it's going to be talking about the extinction vortex and really how important this is. The extinction vortex describes the forces affecting small populations that may cause them to spiral into a vortex, increasingly small populations that endanger their long-term survival, and how that extinction vortex influences the breeding of some of our rare endangered species. And it's absolutely critical at the moment that we look into this to save our species, our rare endangered species and breeds from going extinct. We have to look at the extinction really in a wider context here, really, and look at what's going on out in the world. We're facing the 6th mass extinction, the largest predicted loss of our biodiversity for 65 million years. 1 million species are at risk. 25,000 species are going to go extinct in the next 3 years. And this is not fake news. This is reality. We're staring in the face of these species disappearing. So we have to act, we have to do something, and we have to use science and technology to reverse this cycle. And now, 41% of amphibians, 25% of mammals, and 14% of avian species are threatened of disappearing forever. So extinction happens, just so we know, normally in normal evolution, we have the extinction line down here. This is perfectly normal to happen, normal evolutionary traits where we see extinction happening. But you see this red line, we're in free fall. And this is what is happening now. And we're going to see a lot of these species literally disappearing in front of our eyes if we don't see something. We've seen the David Attenborough films, and this really highlights what is actually going on out there. But the extinction is no longer a term just to the wild species out there and the exotic species. This is happening here and now with us in the UK, with our rare and native species right in front of us. So our rare and native breeds, we're seeing the same sort of population challenges and threats to their survival as we are seeing to the threatened zoo species as well. There's 14 equine breeds, 12 are considered rare, 5 now which are critically endangered with less than 300 females, like the Cleveland Bay, the Dales pony, the Eriskay, the Hackney, and of course we've all seen a lot of publicity surrounding the Suffolk punch. And we have to do something. There is certain things that we can do, but the extinction vortex, how it works is basically the inbreeding, inappropriate breeding, where this is the inbreeding taking place of some of these species due to maybe the lack of numbers or lack of awareness, economic pressures. But this has a secondary effect straight away on these species, where we suddenly get low conception rates, poor quality semen, up to 30% on some breeds, 30%, yes, of foals can have neonatal deaths on foaling or just prior to their due date as well. So we have to do something about it before we start seeing some of our lovely breeds disappearing right in front of our eyes. And it's a lot of this is down to the inbreeding or the inbreeding coefficient, and we can see all the percentages here. When you think sort of 25% is like brother-sister mating, you can see the percentages of all our different species out here. And look, you've got the thoroughbred horse that, you know, Simon Cooper's going to talk about the inbreeding with them. But it's not just the percentages they're at, it's the rate of increase of that. So you might think, oh, you're quite safe at sort of 7% here. But actually, if that level goes up exponentially, you could be in trouble very, very quickly. And this is something we have to be aware of, how quick that increasing is. But there's a lot we can do. There is hope out there. We can use the science and technology out there to try and turn this around and also about appropriate breeding. And you know, Andy Dell is going to be talking about this later with computer generated systems that a bit like your Tinder, a bit like of the horse wheel. We can match these up. But so for appropriate breeding, the Spark system, we can do other technologies. So we can do post-castration epididymal sperm harvesting. We can do sexing semen, genetic preservation, embryo and semen freezing. So there's so much out there that hopefully now that we can actually sustain these breeds. So they're not just going to be here for our generations, but they're going to be here for many, many generations for the future. So as I say, there's lots of new advances out there. And these are some of the ones that we're going to talk through today. One of them is called post-castration epididymal sperm extraction. And this is where we actually take semen from the epididymis from an animal that's just literally very recently deceased, or it's by elective castration, and they send the testicles to us and we can actually extract the semen. We've done about 150 of these equines, and we've done about 45 non-equine species. This particular fell pony that we did quite some years ago, he was having an elective castration, and we managed to get 18 breeding doses of this to save those genetic lines for the future for this particular breed. We broadened this out actually to actually lots of other rare and endangered species. Like here, we've got the mouse deer, incredibly rare. There's only very, very few in the northern hemisphere. So this is one way that we've used this one tool that we use in the equine, and we transferred it back onto other species as well. But these are all the different ones that we've done. The sexing semen. This is where we can home in on the DNA. There's about 4% more DNA in the X-bearing sperm, the female sperm. So if we can home in on those, take those out, segregate them off, this is a way that we can actually produce more females on the ground. This is what we need. This is a short video that we did this year with the first foal born.
And after 11 months of waiting, Ruby has produced a filly foal. And it's not just any old foal. This is a foal born by sex semen. We believe it's the first ever in the world for a rare breed animal to use sex semen. And using these technologies can really help this breed. There's only about 70 or 80 females left in the country. There's only 300 left in the world. They need all the tools of the industry. And just quite incredible now what we can do with science and technologies, and using this technology, maybe, just maybe, we can help stop these breeds from going extinct. But this holds the future. Here.
So that's a combination of lots of different parties coming together, and without all these parties, this just could not have been made. And that's what it's all about, collaboration as well. So the Rare Breed Survival Trust very much helped pull this together. So with Nottingham as well. And this, as Christopher Price said, this project demonstrates the viability of using new technologies to help select female foals to increase the breeding population more rapidly, and achieving through more traditional methods. So this really does show how it works. And the next project we're doing is actually freeze. That was with fresh semen. And the next one, we're actually going to be freezing these sex straws down to be used in what's called ICSI programs, which we're going to show in a minute. And this means we can start spreading those genetics all around the world, all those female sex semen as well. And we can obviously do the male side as well. And this is something we're really, really excited about. We can do what's called oocyte recovery. So this mare passed away, a Cleveland Bay mare, and at Tomlinson Equine, they managed to get the ovaries, take the eggs out, and send them to Italy. And they managed to get 8 oocytes recovered from this method, ending up with 3 embryos frozen down. And that was performed by ICSI. ICSI is called intercytic sperm injection. I think I've said it right. It's quite a long word, but this is really new into technologies. You can see this is injecting 1 sperm cell into an egg. And then on here, this is for the first time ever, this is an actual horse embryo dividing. It's a picture taken every 20 seconds. And you can actually see, in theory, an embryo developing right in front of our eyes. So these new technologies really do play out, and its effectiveness is working here and now. Something I find absolutely fascinating is what we can do in genetic preservation, tissue banking. It's called regenerative genetic banking, using DNA to really regenerate. And this was actually shown on the 8th of August this year. This foal was born, I think I'm hopefully going to say it right, the Brzezowski horse. This foal was born as taking a tissue sample 40 years ago. Their genetic pool was so small, and now they can reintroduce new genetic lines to actually bolster the breed. And at this point, we're actually producing a charity called Nature's Safe, and it means saving animals from extinction. And this is setting up one of the first ever living tissue banks for wild species and rare endangered animals as well. And this picture just shows how it can happen. You can get a small tissue sample from a species or an animal. You can put it in a petri dish, and with that, and you can induce pluripotent stem cells, and you can change those cells into either eggs or sperm in the future. So this really can hold the future, but we do need to store them right now, these samples. So what we really come on to, we stand at a pivotal time at the moment of sustainability and the survival of many of our rare endangered animals, including our rare breeds. So what we've learned from the equine, we transferred onto other species and vice versa. So we have to do this now and without delay to store these and bank these samples down for not just our future, but our future generations. And the time has come to use these technologies to embrace and educate owners and their viability. Sexing semen can really lead the way in getting more females on the ground. And while the latest cryopreservation technologies can facilitate the preservation of reproductive cells and animal tissue samples, the whole genetic preservation for our future. No more. What important this is actually my grandfather here, Bob Matson, in 1936, doing the harvest, and they were our history. They're the ones that fed us during the wars. They're the ones that put food on the table, and it's our duty to hang on to these amazing animals and breeds. And it's our rare and equine breeds are part of, we say, our heritage, with many foundations essential contributing to modern day sport. It is now our turn, and I think this statement is so true. It's now our turn and responsibility to act now and save our rare breeds from extinction. So really, just a big thank you to everybody for inviting me here today to this presentation. It's really, really close to my heart, this. I absolutely love being able to transfer the knowledge we've done to other species. But one take-home message, we cannot wait. We have to act now. to preserve these species. This was some work we're doing in South Africa. We collected off 8 bull elephants, their semen. They're storing this semen and then reintroducing it again to stop this inbreeding coefficient, the inbreeding that's going on in the elephants as well. So we're seeing it right across all different species. So thank you very much.
Thank you very much, Tullis. We've had a number of questions, but several of them are on a theme that we're going to explore in the panel discussion about the role of breed societies, individual breeders, and wider organisations, what they can do and also what should they be doing. So if those who are asking questions don't mind, we can see those on the Q&A, and I'm holding those until that discussion. But one from Bart actually stimulates, I think, which would be very useful. He asked a very specific question, but perhaps I could broaden it. What's the success rate of sex semen as compared to normal semen? You've talked about a range of reproductive technologies. And so, for example, if you were talking about the use of artificial insemination in cattle, and somebody said, is it practical, cheap, effective? You would say it's been used for 70 years. And is absolutely routine, and many farmers do it themselves. If you were saying it in the medical sphere, you would say that it's absolutely routine, as is IVS. So if perhaps you could just go through and start with the basics of AI and embryo transfer. For breeders and breed societies, what's routine, reliable and reasonable cost? And as you get further down the chain, what is more expensive and still more in development? Just if you could sort of go through the landscape of practicality and cost, so that we can get a feeling for what the things we should be doing now that we're assuming are impossible, or vice versa.
Thank you, Tim. Thank you for that. Yeah, I mean, AI has been around now for quite widely now in the equine world. Well, we've been doing it for 30 odd years. So that technique and technology, virtually most vets can do it now. So it's fairly widespread. So it's fairly open to anybody really to be able to use fresh AI, and even most places now can actually do fresh and chilled semen. So in other words, they can, distance isn't a problem to breed mares now. So that makes life a lot easier, and it's fairly inexpensive to do that method. Yes, it's more expensive, you could say, than natural covering, because you've got a little bit more involvement. The embryo transfer side, that just steps up again. The costs, that can all depends how quick that can cost, £2,000 or £3,000, depending on how it's how good the mare is and how good the stallion is, and whether you're using fresh or chilled, frozen. So that's really jumped up quite a bit. The sexing semen, yes, again, all these things, if you have a really good fertile stallion and a fertile mare, you could be looking at maybe a couple of £1,000 to do it. If it's a more trickier stallion, tricky mare, you might have to do more attempts. But it's the value in getting that female foal. That's what you've got to put it back to. How valuable it is to have a female as opposed to a male. And where I think the really gains are is when it comes to ICSI. This is really taken by storm, really, in the equine world, and being able to take these oocytes, these eggs out, and being able to sex the semen and freeze it down and do it this method. We haven't got exact costs yet, but I think this can start to come down massively, because you don't need to necessarily synchronize mares. But yes, there's still certain boundaries we've got to go over, and the more popular it comes, the cheaper it is going to come down.
I would certainly say in commercial cattle production, AI is, almost, when it's used every day, but it's just so routine, people don't think about it.
Yeah,
I would say that ICSI and in vitro fertilization is now looked at as perfect. Embryo transfer, quite a convoluted methodology, was used in the past. More and more that it's absolutely standard. And so I think that's a useful landscape. And there was another question about the costs of epididymal sperm, and I would assume from what you're saying is this is still more in the realm of in sort of full development, but I think the message you're saying is for most of these technologies, don't discount them because they're there or nearly there.
No, well, the epididymal, for instance, so people that want to know the cost, it's about £650 to do an epididymal sperm extraction, and it may be a bit more the more doses you get. But when you put that against maybe bringing a horse in to have that same amount done, there's not a lot of difference. So it's always best to try and get the stallion collected off properly there, and if you can, while he's there, because if it doesn't work, you can have another go. With epididymal, you've only got one shot. Saying that, as a last resort, it's a brilliant way of saving genetics, and we've done this on hundreds of samples now. So it's the epididymal. I'm glad you brought it. It's so important to get out. So many people don't realize it's available. So if anything happens to a rare breed animal, they can get the testicles to a place to get it done within 24 hours. We can actually salvage and harvest those stallions' genetics down. It's a really viable technique.
I know there's a particular participant who has been in this situation. Could you just expand a bit? Obviously, you have a much loved stallion or whatever, but when we're talking about a stallion that is really important to the breed, and we'll come on to that, Andy Dell will explain how that might be the case. If you have a situation, you've talked about 24 hours, are you talking about getting a vet in, taking the, after the animal has died sadly through an accident, doing a, so to speak, post-mortem castration and storing chilled, and you've got a 24-hour window? Is that the, I'm asking about the practicalities as I have in your previous question. Is that really the ballpark we're in?
Yeah, the ballpark for is yes. Ideally, the quicker it gets to us, the better. So ideally, within 12 hours, we have done some up to 48, amazingly. And also the stallion does need health tests as well. So that's the really important part. And of course, the semen has to be stored somewhere else off center until those results come back. So it's really important. That semen is pretty much useless unless those health tests have been taken, because in many years to come, people are going to ask, has the stallion been properly tested? So unfortunately, whatever state the horse is in, it has to have a blood taken off it, and it has to have swabs to make sure that that semen is clean as well.
I'll ask you to expand on that when we have one of our 3 panel discussions is about technical questions. That's an important point.
Yeah. So
I think we're coming to close from your excellent presentation. I'd like to now introduce a presentation from Simon Cooper from the International Studbook Committee, who's going to talk about thoroughbreds and what they've learned from their international genetic surveillance.
Thank you, Tim, and good evening, everybody. I'll be talking this evening about the 3 aspects of the thoroughbred and inbreeding, particularly about the context of the creation of the thoroughbred, the current position of inbreeding within the thoroughbred, and lastly, measures to ensure that the long-term health and welfare of the thoroughbred is looked after. If we just very briefly condense the evolution of the horse into 24 hours, the thoroughbred has appeared in the last 0.2 of a second. So it's a very new breed, and it's an interesting one to study for that reason, from an inbreeding perspective. The start of the breed is very well documented, and I'm not going to go into detail, but the introduction to the General Studbook came out in 1791. And there were about 1,600 mares listed in that first publication. By 1793, so this is only 2 years later, the Studbook was published, the volume 1 of the Studbook was published, and there were over 6,300 mares listed. So this is a breed that's growing very fast. But when we look at the diversification of the breed and the origins of the breed, there are 3 dominant foundation stallions: the Barley Turk, which came from Serbia, we believe, in the 1680s, the Godolphin Arabian that came from Yemen in about the 1730s, and the last one, the Dali Arabian, which has become so dominant, we believe came from Aleppo in Syria in the early 1700s. Now, what is particularly interesting about these 3 foundation stallions is that the male tail line of each of these 3 foundation stallions is carried by only 1 offspring. And in the case of the Dali Arabian, it's Eclipse, and you can see a picture of Eclipse here. And Eclipse, if you look at Eclipse's pedigree, he contains all three foundation sires, and that has become the dominant line in the Thoroughbred. So you're already within quite a short period, in the very short life of the Thoroughbred, relatively speaking, you're already starting to see some loss of genetic diversity. So from over 160 imported stallions, why are there only 3 stallions that carry the Thoroughbred line today? So moving on to what we currently have, what the situation as we currently understand it, we know that breed selection for the Thoroughbred is based on athletic performance. So it's effectively survival of the fittest. We know also that natural cover is the only form of mating in the Thoroughbred breed. There is no artificial insemination. And this was put in place to act as a filter against inbreeding. We also know, as I've mentioned, that 95% of all Thoroughbreds trace their Y chromosome to the Dali Arabian, to Eclipse, through Phalaris, Northern Dancer, Mr. Respecter, and others. And lastly, we also know that it's a closed breed. You can introduce Thoroughbreds that have not been Thoroughbreds into the Thoroughbred Studbook through a promotion process across 8 generations, but effectively it's a closed breed, and we know that inbreeding can only increase when it comes to a closed breed. So we also know that at the current time, the coefficient averages for the Thoroughbred around 0.14. And just to put that in context, around half of the coefficient of the Clydesdale, the Exmoor, the Shire, and the Shetland, and Andy is going to be speaking about that, and Paul also, particularly around the Kerry-Bog pony. But it's more than double the coefficient of horses such as the Mongolian pony, the New Forest pony, the Quarter Horse, and others. We also know that the global population of the Thoroughbred is decreasing. The annual foal crop just in 2005, so 15 years ago, was 123,000. Last year, it was 88,000. So you've seen a nearly 30% decrease in the annual foal crop. And with that, you're seeing the accompanying loss of genetic diversity. And Tullis has already spoken about that. And again, Andy will be covering that. The size of the population is very important. You're also seeing a decrease in the ratio, and this is an important point, in the ratio of stallions to mares. So the number of stallions has reduced proportionately and significantly more than the loss of the number of mares in recent years. And if you just take comparing 2019 to 2020, the decline in brood mares in Britain and Ireland was around 2%. The decline in the number of stallions in that year between 2019 and 2020 was 6%. So in 1 year, you've seen a 4% increase or greater reduction in stallions compared to brood mares. And you've also seen the most number of mares covered by a single stallion rose to 298 in 2020. You're also seeing some stallion fatigue. This is not something that we've necessarily had before in stallion management, to see the stallion through the breeding season with that increased book of mares. And perhaps most importantly, you're seeing an increased levels of inbreeding over the last 3 to 4 decades. And that change has occurred very recently and has coincided with the increase in the size of stallion books from traditionally 40 or 50 or as much as 70 mares to up to, as I've just said, 300 mares to one stallion. And if I just quote a couple of studies that have been done on specifically Thoroughbred and inbreeding, there was a study done in 2011, and I'll just quote a particular passage from that report. It states, the majority of the increase in inbreeding occurred in the last 10 years, for years 1996 to 2006. It is worrisome that the increase is not spread over a 40-year period, but is concentrated in the period following the dramatic changes made to breeding practices in the mid-1990s. And this report is not an isolated report. There are others. I'll quote briefly from a 2019 study that says, striking increase in inbreeding with the greatest rate of change observed since the 2000s. So moving on to what measures we are putting in place, what measures are available to us to put in place. We have accurate 300-year-old pedigree data, which is a huge advantage. We also have some national studies showing individual jurisdictional increases of acceleration in recent years. We also have DNA of every foal. Every foal is parentage verified within the Thoroughbred breed. So we have that vital biobank, whether it be blood or hair. We also have, and again, this is very important, agreement across all the Studbook authorities. And they come under the International Studbook Committee, and there are currently 69 approved Thoroughbred Studbooks across the world. What we don't have, however, is a global comparative measure, an accurate collated global picture from which we can then measure the rate of increase every 3 or say 5 years to see at what rate that increase is occurring. And I think it's important, if I just explain at this point, that the ISBC, or the International Studbook Committee, is the world body tasked with policies regarding the identity and integrity of the breed, but it's the studbooks, it's the responsibility of the individual studbook authorities to monitor trends in thoroughbred breeding, and also to work with their breeder organizations and their breeders to put in place any countermeasures if required and based upon the scientific evidence. So in response, the leading ISBC studbook authorities are now starting to coordinate genealogical data comparisons for that ongoing regular gene pool monitoring. So just speaking briefly about what has happened so far with regard specifically to inbreeding and this increase. There has been, as many of you will know, a restriction in the number of mares in America for thoroughbred sires. They have changed their Rule 14C of the Studbook rules and limited the number of brood mares bred per individual stallion to 140, starting with the 2020 foal crop, who will be potentially breeding in, let's say, 2023. So this will come become effective in 2023. And this is taken from the standardbred breeders who had a ruling in 2009 to restrict stallions, and they have artificial insemination, obviously, within the standardbred breeding industry, to restrict their stallions to 140 mares. And there were some stallions that are inseminating 300 or more mares in a season, which was never thought originally possible within the thoroughbred breed. And that 140 is an estimate. It's not a strictly scientific number. If you look at feral herds, for example, they would be 3 to 30 horses with perhaps 1 to 5 stallions. But it makes a genetic difference because you're allowing other stallions to spread that gene pool wider because they're being bred to more mares, and that effectively reduces that, what I'd call the iceberg effect. So it's important to understand, I think, that the pedigree of a horse is separate and different from its genetic makeup. And the ISBC's position on this is that any study of inbreeding should not be exclusive to either a genealogical study, an analysis, but it should also include a molecular analysis. And we have that science that's emerging at the moment that will provide so much more information. The genealogical analysis will give us, for the most part anyway, that rate of loss of genetic diversity. It comes at a lower cost, and of course the data is available automatically every year once new foals are registered. So it has some advantages. However, it does not provide what the molecular technology can now give us. And this slide just shows what currently microsatellite technology will give us. And next to that, on the right-hand side, you'll see what SNPs, or single nucleotide polymorphism, can give us. So much more information. That can really get down into the detail and analyze every genetic sequence unit of the equine genome. It can also provide underlying genetic mechanisms and explain them, how genetic diversity and inbreeding contribute to either enhancing or harming the breed. And it can also identify genomic regions within an individual horse genome that display higher levels of inbreeding. So you can get into the real detail with this. We can then find potentially a tipping point when inbreeding starts to potentially damage the thoroughbred in some way and affects its health or its welfare and its athletic ability. And I mention that because recent evidence has emerged that a founder-specific inbreeding depression does affect racing performance. It could also answer the questions of, are these inbreeding regions the consequence of selective breeding strategies over several generations? And are these genomic regions linked to a biological function or trait type? So it's two stages of analysis, both pedigree and molecular, and that evaluation should provide the foundation for guidelines and strategies for breed management that are comparable to methods used in other domestic species. But that obviously requires continuous gene pool monitoring, and that is really the most important point, is to keep that genetic analysis going on a continuous basis, so you can start to understand at what rate that inbreeding is potentially increasing. I'd like to finish just on two important points. 20 years ago, the Aga Khan spoke at the Thoroughbred Breeders Association dinner in London, and he warned of the dangers of a small select group of stallions monopolizing the landscape. And he said, and if I just quote him, and you'll see on the slide there, he said, when one stallion can engender up to 300 foals every year, the global inventory of pedigrees in, say, 15 years will present breeders with a totally new problem. In those 15 years, a mare will have produced only some 12 foals on average, whereas a stallion could have sired 4,500 foals. And just to put that in context, that's a ratio of 375 to 1. And that is where that kilter has moved, where it used to be around perhaps 50 or 70 to 1. We've now moved to a much higher scale. That ratio the dial has altered quite alarmingly. Finally, I'd just like to say that in all of this, what we must consider first is what is best for the horse before what is best for the industry. Because if we ignore the long-term health and welfare of the horse, we have no industry.
Thank you very much, Simon. We've got some questions for you. One question probably will include in the wider panel discussion about the role of breed societies and others. But I think there's the common theme which really sort of takes up what you're talking about, the accelerated rate of change of some indices of concern like inbreeding. And you know, I know in some areas, AI, artificial insemination, is a sensitive subject for thoroughbreds, but also what they call increased stallion books, stallions mating with more mares. But also genetic tests, such as tests for what have been called the speed gene. I suppose, and we probably have only got time for really one question. Can you have too much of a good thing with one of those, whether it's too much of a good stallion, too much of AI, if when you're breeding equestrian animals, too much of selection for one gene? Can you overdo it? And will that, for a stud book, and I know Andy Dell's going to talk about it, be a bad thing, overdoing it on male lines and male characteristics?
Thanks, Tim. Yes, I think yeah, the answer is probably yes. I think where the key point is that I think that towards the end I talked about that ratio where you would originally, and when I started in the breeding industry, we had I was working at Beach House Stud, and we had a lovely stallion called St. Paddy who won the Derby. And he would cover at most between 40 and 50 mares, and that obviously has changed a great deal, and that is potentially causing that increase in the inbreeding. But we really need to establish the science behind this, because we don't yet have, we have individual studies across the world, but we haven't yet collated all that information to see, well, what is the picture across all of the thoroughbreds around the world? And that's what we really need to do to get a proper picture, and then continuously monitor that with the science, which Paul's going to be talking about in a minute. With that science, it's becoming much easier. And also, as I think I mentioned, we're getting into some really interesting genetic information or data that we haven't been able to get before, and that'll answer a lot of these questions, because you might have a sire that passes on particular genes that is not a problem, and you might have another sire that it has far fewer covers or mares, but is passing on a particular gene. So there's a lot more I think we need to understand. We're at a very, I believe we're at a very early stage in terms of really understanding A, what is the position, and B, what we can do to ensure the long-term health and welfare of the breed and every breed, actually.
Now that's really interesting and a great introduction to Paul's talk. Historically, in terms of managing a breed, there were really two key components. First was breed standards and breed inspection, and, for example, also performance management. So the performance management in thoroughbreds is racing performance. And then there was Studbook records. However, that has now changed radically with the advent of DNA analysis. Now, it started some while ago with blood group analysis, then to simple genetic analysis, and you'll hear from Paul in a moment about genomic analysis. So it is now, instead of a two-arm, it's a three-arm information group when you're managing a breed and its genetic integrity. So if I could now introduce Paul Flynn from Weatherbys Scientific in Ireland to talk about DNA analysis, and I was very broad by DNA analysis, but he's going to do a lot on genomics. How can it help? And looking forward to hearing his example from a Irish native breed.
Good evening, ladies and gentlemen, and thank you for the introduction, Tim. Also, thank you for the opportunity to present here at the National Equine Forum webinar, whereby I'll provide an overview on DNA analysis and how it can help, specifically within the equine industry. We'll start out with a review or an overview of recent advancements within genomic technologies and how those advancements can feed into better understanding or unraveling the equine genome, and how those extra understandings can build into breed management strategies that can aim to achieve goals and objectives of interested breeds. We'll also take a look at a live working case example of which Weatherbys Scientific have worked collaboratively with the Irish Kerry Bog Pony on a genomics conservation theme project to aim to ensure the survival of the breed into the future. So when we talk about genomic technologies and the equine genome, where does it start? Well, it starts with the equine genome and trying to interrogate that. And that consists of 31 autosomal chromosomes and a sex X and Y chromosome, which also consist of approximately 2.5 billion sequence units. And in short, the more of those sequence units that one can interrogate, the better understanding or resolution of the genetic makeup of the individual of interest one can obtain. In terms of the equine industry, times are changing and how genomic technologies can be rolled out. Simply in that, for the last 2 decades or so, off the back of primary parentage verification requirements, approximately 17 genetic sites were typically genotyped for in your equine individual. We can see where those individual sites are mapped across the chromosomes, and that enabled us to carry out parentage verification, population genetics, and trait mapping. However, as technology within the genomic space has advanced, that technology is now applicable to the equine species. And we can see here, when we take the more recent technology, which is termed as SNP technology, we can look at in excess of 50,000 genetic sites across the genome, where we can see here, based on the red triangles, we have uniform genome-wide coverage, enabling a multitude of applications. On top of parentage verification, we can look at traits, inbreeding, breed composition, and high-resolution population genetics, which can all accumulatively feed into mindful mating advice and breed management strategies. So in terms of a live working case example, as referenced, we have worked with the rare Irish Kerry Bog pony breed and implemented genomic technologies to try and tailor stratified management practices to ensure the survival of the breed into the future. To understand what the genetic composition is of the breed currently, we rolled out SNP technologies in the form of selecting 190 actively breeding Kerry Bog ponies. We took hair samples from each pony. The hair samples underwent DNA extraction. The extracted DNA then underwent genotyping using SNP technologies, whereby we were able to generate in excess of 50,000 data points for each individual analysed Kerry Bog pony. That then allowed us to look at the parameters such as the genetic diversity, genetic inbreeding, how closely related are the Kerry Bog pony to other breeds, how closely related are individuals within the Kerry Bog pony to each other, and then ultimately what is the predicted inbreeding if we were to mate sire A and sire B, sire A dam B within the breed. What would the predicted inbreeding of the offspring be, based just solely on the genetic data without knowing anything about the back pedigree of the individuals? Cumulatively, all that information can be blended into a stratified breeding management strategy. To try bring some perspective on the Kerry Bog pony data from a comparative point of view, we included breeds from two other similar studies, one from the University of Minnesota and one from the University of Aberystwyth, whereby we were able to include nearly 40 other global breeds to benchmark the Kerry Bog pony against. Well, the first benchmarking we did in terms of the genomic data analysis was genomic inbreeding, whereby we were able to establish that the Kerry Bog pony has on average a genomic inbreeding of less than 5%. So on the lower end of the scale in comparison to other breeds such as the Clydesdales, Exmoors and Shetlands, which is to be expected considering their closed and isolated breed nature. We were then able to look at the genomic relationship between breeds based on the individuals that make up each of the breeds within the data sets we had for a group of Western European ponies that we selected. We can see here that the Irish Kerry Bog pony is represented by the yellow squares, each shape or colour representing an individual. The distance between each shape is also indicative of the level of genetic relationship between both, i.e. the shorter distance, the more related they are, the further away distance, the less related they are. Hence, breeds tend to stick together or cluster together. We can see here the Shetlands down the bottom, cluster together in isolation of other breeds, and similarly with the Exmoors within this subset of 9 Western European breeds. The first observation for our 190 Kerry Bog ponies is that there is a nucleus of individuals closely together. And second observation is that there are a series of outliers. And those outliers are moving towards other breeds such as the Welsh A's, and some coming down towards the Shetlands, and also some across to this cluster containing the Connemara's. So first evidence of breed purity, along with evidence of outcrossing within certain individuals in the Kerry Bog pony breed. At an individual level, then we're able to establish the genomic composition of each individual Kerry Bog pony by benchmarking them against the same group of Western European ponies. This graph identifies or allocates colours to each individual. In this case, the Connemara ponies are coloured red, and the higher the amount of breed composition is relative to the amount of colour. So the Connemaras are abbreviated or fixed as red, Exmoors fixed as orange, and similarly Shetlands and Welsh A and D over here on the right. And the colours are linked to the genetic signatures that are associated with those breeds. Our 190 Kerry Bog ponies, we can see that we have a turquoise colour that is associated with the Kerry Bog pony signature. And there is a type triangular profile here, or tapering off of breed purity, whereby we have individuals that are high levels of Kerry Bog pony. And then we also see levels of blue coming from Shetland, Connemara's, the red coming from Connemara's, and also purple from the Welsh A's. So what we have here is a percentage of individuals as high breed purity for Kerry Bog pony, and also a level of outcrossing detected in breeds such as Connemara's, Shetlands, and Welsh A across a number of individuals within the Kerry Bog pony breed. We can then take that at an individual level when we look at anonymized individuals. So Kerry Bog pony 1 had 100% Kerry Bog pony, and Kerry Bog pony 3 had 37%, with the remaining breed composition made up of Welsh A. That enables an understanding at an individual level of what breeding may be required going forward if bolstering of the Kerry Bog pony signature is required or an objective of the breed. Co-ancestry, in terms of understanding how closely related individuals are without knowing anything about back pedigree. So for example, we were able to establish via the genomic profiles that Kerry Bog pony 191 is a full sibling to 114, i.e. they both have the same sire and dam, having a genomic relationship of about 0.5, which means that if you were to mate Kerry Bog pony 191 and 114, they would have a predictive inbreeding of 0.24. Similarly for the remaining other examples here, full sibling examples. Same type profile can be established for half siblings, i.e. that individuals would have the same sire or dam. In this case, Kerry Bog pony 82 and Kerry Bog 41 have the same dam and would have a genomic relationship of 0.24, hence their predictive inbreeding is 0.12, and similar for the remaining half sibling examples. Again, this data can feed directly into breed management strategies at a genomic level. So this data was then disseminated to breeders, where we reported genomic inbreeding coefficients for individual animals. So Kerry Lady, for example, the genomic inbreeding coefficient was reported for Kerry Lady at 2, and so that can be interpreted from a relevance perspective in comparison to the genomic graph we saw, which was less than 5% on average for the Kerry Bog pony. So this individual animal here is not in excess of that average. And furthermore, we reported genomic relationship coefficients, whereby we were able to tell the breeder that if breeder mates Kerry Lady to Kerry Man, the predicted offspring of that individual would be 0.01 in comparison to all other possible sires within the project. We can see that if Kerry Lady was mated to Kerry Mike, the predicted inbreeding would be higher. So that individual information enables the breeder to potentially steer the level of inbreeding in future offspring for the breed. Key considerations: genomic technology paradigm change is on the horizon for equine. Ultimately will enable us to analyze more genetic sites, will enable a multitude of further data application types. The greater the uptake to SNP technology enables potentially more cost-effective solution or comparable to the current 17 microsatellite marker systems. We saw how the technology works within our live working case example, in this case, a conservation genomics team project, whereby we now understand the Kerry Bog pony breed does not have an overall average inbreeding problem. We can see recent cross-ancestor events for breeds such as the Welsh A, the Connemara's, and the Shetlands. We now understand the individual genomic relationship between pairs within the breed and how that can help us predict inbreeding on future offspring, and which can all accumulatively feed into a breed management strategy for the breed. Future considerations: this toolkit is applicable not just to Kerry Bog ponies, but also other breeds, equine breeds, and not just equines, but other species such as bovine. This is all genomic data, which can be blended with the type data that Andy is going to talk about next, pedigree data, and accumulatively, both genomic and pedigree data blended will enhance the accuracy of those data findings. How these strategies are then implemented is of obvious concern too, and that is critical in how the data is used. And again, Andy is going to discuss practical manners of which breeding strategies can be rolled out. Continuous monitoring is required, as Simon referenced within the Thoroughbred inbreeding piece. It is just a snapshot or point in time that requires profiling across a series of data points. And that profile will then better educate breeders what changes need to be put in place, if any. That's it from me. So thank you very much for your time, and happy to take any questions.
Paul, thank you very much indeed. One thing I wanted was perhaps follow up in a general way from what we talked about with Tullis, and not put you in a position where you're sort of quoting for services. So perhaps use a parallel example. I know we'll never probably get the economies of scale in cattle, but in cattle breeding, commercial cattle breeding, can you give us a flavour of the routine nature and the low cost of advanced genomics as being used today, just to give the equine community of a little bit of a vision for the future of my understanding how routine and cost effective it is when it is widely applied?
Thanks, Tim. Yes, look, a very good question. And for sure, the equine industries is not quite there yet to leverage economies and scale within this technology. And the cattle world is a great example. How the technology can be leveraged when economies of scale are achieved. And what you typically see within the cattle world is that this technology that I've described there, being able to look at nearly 50,000 genetic sites at each individual level, is very feasible. It's feasible at an economic and commercial level, and it's common practice within the cattle world now. And being able to generate that type data allows the multitude of applications that I outlined there in my second slide, in that we have a position where when we genotype an animal, that the initial remit of that genotype is to obtain the 50K profile, which will enable parentage verification. And not only parentage verification, but it can allow sire or dam allocation at a very accurate level without knowing any nominated sire or dam, if those sire and dams exist in a database when you have 50K data. That then can be built on in terms of being able to granularly map traits in tandem. So you can have 50K profiles that provide that genome-wide coverage, but also target very specific regions of the genome, whereby those regions of the genome have an annotated effect. So for example, a myostatin gene, which will code for muscle mass, or another region of the gene that will code for whether an animal is born with horns or not, so polled. So that type testing can be all done in tandem with that one genomic profile. Furthermore, then, the likes of inbreeding can be in tandem mapped. But the real commercial driver within the cattle world is genomic selection. So because you have such genome-wide coverage and associated phenotype data, which brings real value to the genotype data, you can then establish genotype-phenotype associations of which particular traits of relevance to the breed from a commercial aspect can be selectively bred for. Not in a selectively sweep type manner, which can cause potential inbreeding or bottlenecks in the genome, but traits can be bred for and yet be mindful for the surrounding genome and ensure that there is no loss of genetic diversity. So that's the type of granular mapping that, Tim, that the cattle world is experiencing and how they're leveraging it from a commercial perspective.
That's really, really interesting, and that you're getting some comments of, well, actually one of the comments simply sums it up. Wow. So real opportunity. Let's go now to Andy Dell, and he's been working very hard and changing behaviours and getting things done in a particular breed, which he's going to talk about. So he's going to bring us back to the daily business of breeding, but actually how then he's applied genetic programs, which do reflect the DNA, to improve breed diversity. So Andy, over to you.
Well, good evening, everyone. For those of you who don't know me, my name's Andy Dell, and I've been involved with Cleveland Bay horses for the best part of 30 years. And what I'd like to do this evening is to share with you a breed management program that's been in use with the Cleveland Bay Horse Society for the last 16 years, and to show you the benefits that it's been bringing. And that breed management program is known as Sparks, has been adopted by Cleveland Bay Horse Societies around the globe. But that owes its origins to the world of captive wildlife management and computer software that was designed for managing captive wildlife populations in zoos. But from our perspective, the project owes its origins to the digitization of our Studbook back in the 1990s. And what that revealed was a concerning increase in inbreeding. Inbreeding that had been increasing at a linear rate, but such that by the year 2000, it had passed through 20% and was marching on to 22%. And if I tell you that 25% inbred is the equivalent of a brother-sister full sibling mating, that rings alarm bells. But our previous speaker, Paul Flynn, spoke to you about the use of genomics in managing Kerry bog ponies. And we have to ask ourselves, at a time when genomics is increasingly accessible, is the use of pedigree still appropriate? Well, in 2017, a group of researchers looking at the Australian thoroughbred population found that where a pedigree is really robust and really deep and really complete, there's a very good correlation between pedigree-based inbreeding and genomic-based inbreeding. And what Cleveland Bays have in common with the thoroughbred is a really deep, really robust, really complete studbook record. So yes, where we've got that complete record, there very much still is a place for pedigree. So if we want to manage our endangered populations, we've got a number of choices. The first of which would be to take what we've got, divide it up into subpopulations and keep them isolated from each other. But that brings its own problems, because it means we could have no sale and exchange of live animals, no exchange of genomic products, and that would compromise the breed. It would subdue the market, so it's not an option. Then there's random mating, which is a recognized strategy, but it needs to take place under idealized conditions. But we don't keep our equine populations under idealized conditions. We decide what mares mate with what stallions. We provide fodder in winter and we call the vet when things go wrong. So random mating isn't an option. And then we come to managing inbreeding, but managing it indirectly through control of mean kinship. And that's how Sparks works. You've heard about inbreeding and mean kinship earlier this evening. Inbreeding, the relatedness of an animal to itself down both sides of its pedigree. And mean kinship, the relatedness of any one animal to every other animal in the population. And from a breeder's perspective, Sparks is based on data sheets, whether they're mare-based or stallion-based, and I've got both up on the screen behind me. And these are similar to the sheets that Paul Flynn showed you for the Kerry bog pony, perhaps a little bit more sophisticated with the colour scheme. They've had 16 years to evolve. But what these sheets show is the mean kinship of every male and every female in the population, but more importantly, the co-ancestry, the theoretical inbreeding coefficient of every possible male-female combination. And then we've got a colour scheme which helps guide breeders towards making appropriate choices. And that traffic light scheme is divided up into 4 groups, 4 bands of matings. First of all, we've got the green matings, the ones that we want to encourage breeders to do. And the reason we encourage them is because these matings mate animals of very similar mean kinship, but produce progeny of lower than average inbreeding for the breed. And what that does is it retains as much diversity as we can for the next generation. And it also means that we've got the maximum number of green matings available in future generations. So we want to encourage these types of mating. But for whatever reason, those matings might not be possible, whether it's economic or geographic reasons. So if not, we encourage breeders to look at the yellow band of matings. These are the best of the rest matings. And these still mate animals of very similar mean kinship, but they produce animals of higher than average inbreeding for the breed. Higher than average, but lower than a maximum threshold that we set. And in the case of Cleveland Bays, we've set that at 24%, bearing in mind that 25% is a full sibling mating, or the equivalent of mating a stallion with his own daughter, or a mare with her own son. Then we come to the orange matings, and these are matings that we would rather breeders didn't do. We want to discourage them, and the reason we discourage them is because they bring together animals of very dissimilar mean kinship. They mate animals carrying the least common genes in the population with the most common genes in the population. And the problem is, if you do that, there is almost no way of separating them out in future generations. And it also reduces the number of green matings, the options for breeders in the future. Then we come to red matings, and these are matings we really need breeders to avoid. And the reason we want breeders to avoid them is because these are highly inbred, highly damaging matings. Highly damaging because they increase the chance of deleterious genetic traits being expressed, these harmful genetic traits. And they also have a very negative effect on something called effective population size, which is something Tim Morris talked about at the start of this series of talks, and something that I'll explain in a little bit. But we can't breed by numbers alone, and I like to think of Sparks as just one tool in the breeder's toolbox, which divides itself up into two types of skills. First of all, we've got skills that deal with phenotype, the makeup of the horse, things we look at with the horseman's eye. Do we like these horses? Should we be breeding from them? Are they of breed standard? Have they got a leg at each corner? And then there are things that deal with genotype, the genetic makeup of the horses. And in the past, what we might have done is take the stud books down from the shelf and shuffle around pedigrees on pieces of paper on the kitchen table. The problem is that when you've got 30 generations of pedigree, you cannot do that. There are over 1 billion animals in the 30th generation. So you have to resort to computer analysis. So Sparks is just another tool in the toolbox, but what it does is it leaves breeders in control, but knowing that they can make appropriate choices, not just for themselves, but for the breed as a whole. They can help to avoid these highly damaging matings, and they can also make sure that what they do leaves as much of the genetic diversity that we've got for future generations. It also means that we can identify and prioritize mating from animals of lowest mean kinship, the ones carrying the rarest genetics, that we can breed from as many males and as many females as possible, and that we can avoid the overuse of any one or any two stallions, which is an inherent problem in equine breeding. I showed you this graph a little bit earlier on, with inbreeding increasing past 20% towards 22. Well, since the advent of Sparks, inbreeding does continue to increase, but it increases at a much reduced rate. It's not zero, but it's getting close to zero. And what that in turn has done is it's had a really positive effect on effective population size, which if you look at the graph behind me, I've drawn a dotted line in at 50. 50 is a magic threshold below which breeds are not seen as viable. And in the 1980s, Cleveland Bays took a dive in the wrong direction through that barrier. They reduced down such that by the 1990s, the effective population size was below 20. Well, with the advent of Sparks, we've turned it round. We pushed it back in the right direction through 50 in about 2012. And it's carried on increasing such that by the end of 2020, it was at 147. It hasn't been all smooth going. You can see that there was a big blip. It went backwards for 3 years between 2014 and 2017. That was as the result of one breeder carrying out 3 really highly inbred matings year on year on year. But overall, it's been extremely successful. And we've learned a lot of lessons. What we have learned is that where there is uncoordinated breeding amongst close populations, you do get this substantial accumulation of inbreeding. That is highly damaging, with a high chance of these deleterious, damaging genetic traits being expressed. And also it has this really damaging effect on effective population size. But with the use of a combined strategy, we can do something about it. We've seen how it's brought benefits to the Cleveland Bay breed. Well, under the umbrella of the Rare Breed Survival Trust, we've managed to expand the use of Sparks. It's now in use by the Eriskay Pony Society, and it's in use by the Suffolk Horse Society. It's very early days for these breeds. With the Cleveland Bays, it's been running for very nearly 2 generations, and we can show that there's been a really proven benefit. With these, they've been in use for 2 or 3 years, so it's very early days. But what I can say is it's definitely brought benefits to the Suffolk horse, because you can see in the centre a picture of a purebred filly foal that was born through the use of sex semen in 2020. Well, that mating was selected using Sparks. It was a green Sparks compliant mating. So we know that that filly foal carries genetics that are really helpful for the future of the Suffolk horse. So I hope I've shown you that by using pedigree, we can do something really positive for the breed. We've shown that where breeders work together with breed societies, even though these breeds are endangered, they do have a future. So thank you.
That really is a telling example to the key question, can something be done? And I'd like to focus, we've had a lot of questions here, but there are some key themes on what breeders, breed societies do and international, but I'd like to focus, and we have probably just over half an hour, so I'd like to spend probably just less than 10 minutes on each topic and give us a little flexibility to catch up on anything else. So I'm going to start actually with probably just a brief exchange between Tullis and Andy on technical issues before we get down to the key role of breed societies and breeders and then international. So the focus really of my two questions to Tullis and Andy is that there's a lot we can do. The technology is expanding every day. There are some restrictions, and for example, Tullis talked about the reason for the necessity for health controls when moving genetic material around the world. There are, let's call them cultural issues. For example, there is now a thing called the NAGOA protocol, which makes sure that you don't take genetic material from vulnerable communities, basically steal genetic information. There's a question over whether there is an infrastructure for tissue banking, and there are ethical questions over cloning. Not all in all countries, but in some. But things are changing. We've seen a debate this week or in the last 2 weeks about gene editing in plants and livestock from the Secretary of State at DEFRA. So I think that I'm going to ask the questions together so you can see the context. Tullis, AI has been around for a long time, and forms of embryo transfer, and you've talked about ICSI in particular. Over the next decade, what do you see becoming routine? And second then to Andy, does that call you any concerns, or is it not so much the technology, but how it's managed and controlled is the main issue? So are there any inherent issues with particular reproductive technologies, or is it all down to having a traffic light system for everything? So those two questions, and if we could be fairly brief, because I think we've got a lot of questions on the role of breed societies and their members. Probably Tullis first. Where do you think it's going?
Just on technologies, yes, as we see, I remember when we started AI years ago, it was sort of slightly, well, we're messing with nature now. Now it's the norm. So things are moving on so quickly now. So I really can see ICSI playing a big part in the future. And one thing I didn't mention, obviously, before we were talking about epididymal side as well as storing those stuff. The tissue side, tissue banking, I can really see the Natural History Museum are really getting into this as well, and seeing biobanking, storing these samples down so they can be reused. And it's not 24 hours for that, it's 5 days. So if a certain animal was to pass away, we've got 5 days to get those samples in to freeze them down. So, and that's starting, I can see that's going to be a new way of preservation for the future. But when it comes to reproductive techniques, yes, ICSI now is getting going, and I can really see, and hopefully sexing a semen alongside that, put those two together, this can have huge benefits for the rare breeds.
So Andy, that if you could make some comments, and for example, it's really interesting. One of our questions was the role of sex semen in international breed conservation. So things that worry you and things that encourage you, really.
There's actually very little that worries me. I think the really important thing is that breed societies take advantage of the ability to monitor and understand their populations, where they are at this moment in time, and can plan where they want to take them in the future. Things like having a proper mean kinship analysis so that you can identify animals that are really high priority breeders because they're carrying the rarest of the rare genetics and the genes that are most at risk of getting lost. When breeders and breed societies are considering what animals are important for gene banking, I know there's a lot done with the Rare Breed Survival Trust and the regeneration scheme, you know, to identify the stallions that actually, yes, you want to profile right across the breed to be able to recreate it for the doomsday scenario, but you also want to make sure that you've collected the animals of lowest kinship. And then when you've got your gene bank, then what you need to be able to do is to go, okay, if I add this next stallion, is that going to make the mean kinship higher or lower? And you should be aiming to make it lower. In terms of other technologies, the ICSI, yes, you know, it is novel, but it is doable. I know Tullis and I were stood on the yard at West Kington last year when things went very pear-shaped for one Cleveland Bay breeder, but we were able to salvage something because we were able to identify that mare as a high priority breeder. We were able to use sparks to get semen from the RBST gene bank that was A, exportable, and B, it was a green sparks match. Yes, at the time it had to go to Italy. I know things are changing rapidly, but those embryos are now back here in the bank and hopefully a resource for the future. So I think it's a really exciting time, and it's understanding what you've got and what you can do with it that's the important thing.
Andy, thank you. That was an excellent introduction to probably what is the really meaty panel issue here, because many of the questions relate to this, which is what should breeders, breed societies and international organisations do? I'm going to focus domestically first, but then go on to international later. So what can breed societies and their members do to avoid inbreeding, extinction vortex, manage effective populations? How can the Studbook manage genetic diversity? How can it deal with, one of the questions was, the high incidence of cryptorchism, a genetically based disease in Eriskes? Is there anything in wider government regulation that is relevant about breeding practices which could be said from the presentations we heard tonight be outdated? How do we change breeders' behaviour to look at, not drive through the red traffic light, to use the analogy that Andy used? So I think the first thing I would say is that in the resources, I have placed something that I would probably put as a constructive challenge to breed societies, which is early this decade, the Farm Animal Genetic Resources Committee, chaired by our very own Tim Brigstocke, produced a report. And there was some guidance at the end of that, and that included that breed societies should have a breed conservation committee. Now, many breed societies have show committees and finance and governance committees and fundraising. How many have breed conservation committees in which they look at regular updates on coefficient, inbreeding coefficients, the effective population? They look at signs of genetic disease and how those could be addressed. On the NEF website, you will find a link to a model template for a breed conservation committee. And certainly that's something the Rare Breed Survival Trust has been thinking, how we can help breed societies, you know, working together with best practice from FANGA, the FANGA committee, to develop those, so at least they know what the situation is. It's really interesting, coming to regulation, before I go on to just ask Andy to expand a bit on this, that in the agriculture bill, which is about farming, they talk about both native breeds getting financial support, but also equines. But is this going to be, and this is because of the public good of their conservation and biodiversity, but is this going to be a free ride? Will the zoo technical legislation say, actually, you need to have a breed plan, not just focus on the sires I like? Because that, to me, would be quite reasonable. So perhaps the first thing for our panel discussion is, Andy, from what you've learnt, good and bad, from the Cleveland Bay experience, what would you say to other breed societies?
Well, I think the very first thing is that it's as much about managing people as it is about managing horses, and we need to educate and get breeders on board and pulling in the same direction. But there is an onus on the breed society that actually holds the data and maintains the stud book to analyze that data, to see where they are at this moment in time and what's the history been. You know, it's only through digitizing our stud book that we were able to see not just that inbreeding was bad, but how rapidly it was increasing. Tullis said very early on, it's not just the level of inbreeding. You know, at Cleveland Bays are at 22%. We can't magically make that go away. But what we can do is control how much it continues to increase. So breed societies need to get their data to a position where it can be analysed and meaningful and understandable reports can be produced. That was difficult 10 years ago, but now we can do that. And I think that both the Rare Breed Survival Trust and DEFRA ought to perhaps be looking towards helping breed societies facilitate that. Then once you've got that data, then yes, we've shown both through genomics and through pedigree based means it is possible for breed societies to put very, very workable breed plans in place and actually to get good results. But then the monitoring is really important. You know, we can show unequivocally that Cleveland Bays has improved, and we can say where it is at this moment in time. But we need to continue that monitoring. As Simon Cooper said, it's not just about getting it sorted today. We need to carry on that monitoring. We need perhaps, if we're managing through pedigree, to do regular checks using SNPs just to validate that you know, genomic inbreeding isn't accelerating at a greater rate than pedigree-based inbreeding. But once we've got that monitoring in place, we can actually see where breeds are and help them get back above that 50, stay above that 50, and you know, report through to DEFRA that things are going in the right direction, so that in turn DEFRA can report back to the Food and Agriculture Organization of the United Nations, and ultimately that's you know, the public good that breeders ought to be rewarded for.
I mean, it's interesting you say public good, because many breed societies are DEFRA recognised, and, you know, is that a free ride? And many are also registered charities, often to improve the breed X. Are that, you know, my challenge would be to them, is there enough focus on that? But let's turn to, so to speak, the other end of the spectrum, to Simon. You know, the thoroughbred breeding business is not a charity, so some owners and breeders may say it is. You know, what are the drivers, you know, perhaps business and financial and reputational, that you think are going to drive the thoroughbred industry to address this issue?
Thanks, Tim. I mean, I'd endorse what Andy says, which is that it's really about, it's as much the people as it is the horses. And I think what we need to do is, particularly on the people side, is be completely transparent and keep that communication between the breed societies and the breeders and any of the other bodies involved in who must have the best interests of the breed at heart. I think it comes down to what Paul was saying. It really comes down now to the science. We have really, for the first time, some fantastic tools in the armory now, which we didn't have even 10 years ago. And I think that has given us such a good understanding, perhaps too much understanding, in that we now, a bit like climate change, because we've now got the science, we understand the dangers ahead. We're not blindly wandering into it. And I think it's incumbent on the breed societies talking to the breeders and talking to all the other agencies involved to keep that dialogue going, and to keep, again, as Andy said, to keep that continuous monitoring going, rather than simply doing, well, we've done the report and everything's fine, and and that's it. I think it's a question of having a plan, a long-term breed management strategy or plan in place that takes you. I mean, we want to see the thoroughbred around in the same healthy position we believe it's in now in 100 and 200 years time, and it's up to us to ensure that now, rather than waiting for the next generation to do that.
And that's very useful, and I'm just going to open it up to the other members of the panel if they've got any comments, but just to make sure we're addressing some of the comments coming through, it's about looking forward rather than back, and things like, if let alone oocytes, the female gametes, but actually fertilized embryos, if they are the future, should we be storing those just as much as semen? And should we be actually using technology better? We've learned a lot, and we've been hearing a lot about customs and forms and delays in the last couple of weeks, but I'm sure many of you have used Amazon One Click or eBay Buy Now, and it just happens in the background, just like the traffic light system. So whilst, yes, there's a lot of technology and science behind it, I do think we are in the stage where there can be some really useful practical tools that help breed societies move forward on the conservation of their breed. So just looking at Paul, you already mentioned that the costs are coming down, whether you or Tim wanted to make any final comments. Tim is also a past chair of the Rare Breed Survival Trust. Is there anything that either of you would like to sort of add to this whole question, that actually it's not about the technology, whether it's IT or it's more about people and helping them understand what's really best for their breed? Any comments from Paul or Tim? Tim, and then perhaps Paul.
Yeah, I mean, I agree with what's been said. I think the key thing for breed societies in their conservation efforts is about science into practice, rather than groups of informed enthusiasts who are not quite sure what they're doing. And that's a sea change that I think has got to happen. I think, as far as you, Tim, you've mentioned a number of times the role of government. I think government will provide pump priming money to bring some of these things about, but they'll expect the industry in its broadest terms to actually pick it up and run with it. And I think the key thing is about how you implement science into practice.
It's really interesting you say that about government being an enabler. We had a question about grazing, and this applies to whether it's Dartmoor, Exmoor, Fells, Bodmin, and there are breeds associated with all those sites. But actually, it's not so much government telling people how to do it, but if there is government support, is it not reasonable that if biodiversity needs within the breeds need to be preserved, and that is a condition of the support? Paul, I tried to make sure that you weren't put in an invidious position about costs and everything, but really one of the things we often say now about IT is you're only limited by your imagination. Do you think the future's bright for practical, cost-effective tools for the equines? Let's home in a bit on that.
Absolutely, yes. The technology and how it's progressed in recent years, it's there to be leveraged. We used that term previously, economies of scale. will be the ultimate driver here in terms of adoption. Obviously, cost is a big key. But I think in the early days, what is going to really drive it is for breeders to understand the value add that this technology can bring. That is the immediate demonstrative gain that the technology can bring to industry to achieve goals and objectives of, let's say, a rare breed. And the rest will follow in terms of adoption, and specifically within the equine industry and enabling this technology that I referenced to, SNPs. There are several governance changes on the horizon, particularly around options to carry out parentage verification, which currently under the recommendations of the International Society on Animal Genetics, microsatellite markers is the recommended marker. So obviously there's a big drive towards generating microsatellite size genotypes based on that recommendation. However, there is an expectation this year that ISAG will now recommend SNPs as a molecular marker to carry out parentage verification. And with that, it will be nearly the first stepping stone for the equine industry to move from microsatellite markers to SNPs and bringing more volume to the game. Hence, the economies of scale, being able to leverage the technology will come good too. So that's a huge piece that all of a sudden we could find ourselves with the equine industry being able to leverage this technology and make the future bright, as you put, Tim, in terms of trying to address any challenges that breeds may have. But just, I suppose, also just to reference the fundamental challenges, even outside of the technologies that we referred to there previous, even in the cattle world, it's the basic principles, as my previous co-speakers have said. It's the dialogue, it's the communication, it's the continuous monitoring and being able to demonstrate to breeders the value of the technology. And within the bovine world, some of the most fundamental challenges is around ensuring that there is a consolidated society overseeing the goals and objectives of the breed. And that it's just back to that communication piece. And once that can be kept tight, and that society updated, good and all as the technology is, without the society, the technology is aimless. So it's about consolidating it all, and not look. Tim, you've referenced cost there once or twice, and not want to avoid the question, but for sure, with the proper economies of scale in place, the equine world can leverage something comparable to the bovine world. And to establish a 50k genotype, you're looking at somewhere at the moment now, it could be 150 euros, but we'll quickly get to less than 50 euros a sample.
It's really interesting because we've had some questions about costs, and I think the message you said is, yes, I mean, it's not nothing, but actually the more we all do it, the bigger the global and cross-breeds economies of scale, and that answers probably the most frequent one of the questions just been raised. So I'm now going to come to international aspects, because we've had questions about setting up national breeding plans locally in individual countries to avoid contraction of genetic diversity, how to work with a restricted gene pool, what's best practice, international exchange. And I wanted to give an example, and there is some experience from cattle, which I think is a very good example, and it also spares blushes between any equine examples. In cattle, they have said that Britain has been the stockyard of the world in terms of Hereford and Aberdeen Angus and beef cattle around the world. And so, our cattle breeds went out and populated many, particularly Commonwealth and South American countries. And one particular breed society was very dismissive of the genetic value of some of these. But actually, a small group of enthusiasts within the society went and looked at the genetic purity of a particular breed in Australasia and found actually it was purer and more useful than some of the rather diluted UK versions. And they did that using genomic technology, exactly what you were talking about, Paul. And so I think there are some real opportunities there. So really the question I had was, how do we set up international breeding plans? So for example, and I'm thinking particularly here of the heavy horse breeds, and coming to you, Andy, that we actually critically evaluate the international breeding populations. There is an international stud book committee for thoroughbreds for the independent national stud books. Do we need, for example, a particular heavy horse breed, to see what we can learn from that, to see what the worldwide genetic pool can do? Because are we being narrow-minded and with forgiving the rest of the UK nations? Are we being little Englanders about our breeds? Are we missing genetic diversity around the world? Start with Andy.
Yeah, really pertinent, because if you look at that mare that's looking over my shoulder right at this moment, she's part of what is already a global stud book, and Sparks has been set up based on that global stud book. There's a bit of a conflict because under the old EU Zootech regulations, perhaps Cleveland Bay Horse Society should not have been registering horses from outside of UK and Europe, but it has been. I know it's amongst 24 other livestock breed societies that have been guilty of doing that. But actually, that's probably been the saving grace for this breed, because we've been able to look at the global diversity that's available. And the Spark sheets, yes, it's categorized. I noticed somebody flashed up a question asking about how we deal with deceased horses, that the Spark sheets deal not just with horses at home and Europe-wide, but overseas as well. And it also does deal with horses that in particular have had semen collected but are now deceased but are still available. So global stud books are really important. I know on the resources, I asked for a link to be put up to a website, breedingfordiversity.com. That's by a gentleman called Pierre Olihek, who his specialization is in canine biology, but all the messages he puts in there about how you should look at and how you should manage your breeds are exactly how we've set up Sparks. And he talks about globalization. I think it's really, really important that we take, these are global resources. In terms of the heavy horses, well, yes, I've been working with the Suffolk Horse Society, and they do have an American Suffolk Horse stud book, which is separate from the home stud book, but there are moves afoot to start to incorporate that data into a Sparks analysis. And I think if we combine pedigree and genomics, and we can have a look at what integration, what admixture there has been, we will be in a really good position to use them as resources. I know I talked about dividing populations up and keeping them separate ad infinitum so that they can develop on their own, but perhaps with some of these breeds we're at a point where it's time to have a look at what there is overseas and start to bring things back. Certainly in the Cleveland Bay world, I've been asked the questions a number of times by UK breeders, what semen would it be most advantageous to re-import from North America? So we need to think along those lines, definitely.
So we have several questions about stud books and Brexit, but I think it's fair to say that this is about mutual recognition rather than a temporary hiccup over, and that's which has been resolved about mutual recognition of stud books. I'd like to broaden it, and at the risk of sounding like Boris Johnson, think not just about the EU but globally, and ask Simon, do you actually think those breeds with significant international populations should have something like the International Stud Book Committee for their breeds? Could you operate global thoroughbred breeding without it?
It's a good question. It works very well. The International Stud Book Committee was set up in 1976, and it was to put in place basically agreement across all of the approved stud books to have some form of accreditation to ensure that all the stud books were working to the same standards around identification, DNA verification, and all of those things. So there was a consistent standard across the world. The way that it works now, and I can see it being expanded potentially around this particular topic, is we have the 10 largest stud books around the world sit on the committee, and then there are 14 regional reviewers who are there to assess and review other stud books within their region. There are 4 regions: there's North America, South America, Europe, and Asia, and Australia and New Zealand. And Asia and Australia and New Zealand is one. The largest area is Europe. We have about 35 thoroughbred stud books within the European region. That's not the EU, that's the wider European region. And that works very well. And I think it certainly, in terms of the monitoring of inbreeding or any issues, deleterious impacts of breeding that could occur or are expected or anticipated in any way that need to be monitored, could come into that regional reviewer remit potentially in the future. It's a very good structure. It's been there for a long time, and it certainly, in terms of, without it being in the strictest sense regulatory, there is a concordance, if you like, across all of the stud books, and they work extremely closely together. Also, I should probably mention very quickly on the international front is we do have not a huge number, but there are shuttle stallions, as people know, moving across hemispheres, between North and South hemispheres, so that you've got this double breeding season for some stallions. And at the very high end, you've got mares coming either going down to the southern hemisphere or coming up from the southern hemisphere on the non-breeding 6 months of the year for those stallions. So there's quite a lot of diversification that way. And interestingly, there was a study done in 2019, I think it was 2019, which identified the highest inbreeding coefficient amongst thoroughbred populations was in South Africa. And I would, I'm not a scientist, but I would hazard a guess that was to do with the fact that they don't have a lot of diversification because of their isolated position because of African horse sickness.
No, thanks very much. So really, a question now probably for Tullis to not give a sort of complete answer, but just sort of muse ahead to some thoughts he's been having on this international aspect. And again, I don't want to sound like Boris Johnson, but we are master our own destiny. We can set our own zootechnical policies now. On the issue of native breeds, we have populations around the world. Do our regulations, whether they are for the newer technology or health regulations for the older ones, such as health regulations for semen and things like that, do you think that they are suitable at the moment, or could be enhanced, or rather should be enhanced, or should be simplified in the past it's been a big focus on health status, but actually there's no point in worrying about the whole health status if the animals that existed. Are they suitably balanced with allowing international biodiversity? Good challenge for you, Tullis.
Yeah, I think the most important thing we've got to see is the biosecurity of our country. Have to admit, is one of the most important things, without doubt. So moving samples around the world, it has to be properly tested. Otherwise, we are in a fortunate place. We are an island, and there's been some huge outbreaks of EVA and other diseases. And we've just seen in our own pandemic, we are seeing now how the movement of people is making this disease move. And it's the same with movement of horses or horse products. So first of all, yes, I think it would be great to have a more uniform standard all around the world, but we've been looking at that. It's very, very difficult because different countries have different priorities and different diseases which they should be testing for. And it would be nice to have a more generic, the main testing to be done. So that means the semen can be shipped to virtually all countries around the world. But life is not like that, unfortunately, and each countries have a different health testing regime. But it would be great if it was more unified, so these samples could be moved from one country to another a lot easier. I just wanted to bring up one point that I didn't bring up earlier. We've just saying how the issues are happening with the quality of semen, things that we're seeing. We've just had a dissertation paper that's just been published from us, and it was looked at did 11,500 collections. It looked at over 20 years. And this is quite staggering, this is, and where we're going. And it looked at, and this is all the ones that are done here. So I've done on this roof, of over 1,000 different, it was 1,036 different stallions. And they noticed that the amount of semen they produced was the same. Actually the volume was about the same, but the quality dropped by 10% over that 20 years. And that's quite staggering if you're looking at, and we're not talking 10 stallions here, we're talking 1,000. So it is so important that we look at what we don't exactly know why this is. Is it inbreeding or is it the environmental changes? So we've got to find a way of obviously looking into this more, because in another 10 years it could get worse. So going back to saying about bringing in new genetic lines and new strains to maybe bolster these breeds up, I think is so important from all over the world to try and introduce new genetics into some of our rare breeds. You look at the Suffolk Punch, they're flourishing, or not, I shouldn't say flourishing, but they're doing very well, I should say, or better than us in America. And maybe we should be looking at them and bringing some of those genetics back in here to strengthen and bring down the inbreeding coefficient on some of our breeds. So I think, yes, we can do it, but we have to look at the biosecurity, because what we don't want to do is just be bringing something in that hasn't been properly tested. Well, we can't do that anyway, but it's really, really important that our biosecurity is looked at as well.
Well, in fact, we could do it, but actually your message is that would be a false economy. And actually the message I'm picking up from the other participants is almost a less is more. Make sure that if you're moving, and it will probably be genetic material, less animals, that it isn't based on some I like that particular line type thing, but a proper analysis based on pedigree, genomics and others, so that actually these important hurdles of health and others, which do cost something to the, is that you are getting value for money in these expensive transfers. But I think the message is these expensive transfers are very important. It's a genetic resource both ways that is being underutilized, whether in big stud books or smaller stud books, and that I think is a very clear message. Are there any challenges? And I just wanted to ask you we haven't really talked about the storage of semen and eggs and embryos. Perhaps I'll ask Tim this, is in terms of the big picture, international coordination of this, good, bad or ugly? Is it done well? Is it done a little bit, or is really not being done and we're missing a trick here?
I think it's been done, but it's a bit piecemeal. It's not terribly well coordinated, and it probably could do better. Just going back, and the number of the questions have come up, Tim, about Brexit, and I've been, as the chair of the FANGA committee, I was actually on a call today, because of the 55 approved PIO stud books, 27 of them are daughter stud books, and not all the mother stud books are in the EU. So there is an almighty problem that we've got to resolve, and it's really not very easy to see how we're going to get through this. But we will keep all Breed Society people informed when things are happening. But government is well aware this is really quite a difficult issue.
Yes, and the issue there is that you can have the best genetics, but if for a legalistic reason it can't be recognised as that breed,
yep,
it can confound those best intentions.
Yep.
I'm going to just actually, rather than doing a summing up, I'm simply going to say I'm just going to go to the four speakers who put in a lot of effort. Is there any final thoughts, and you don't feel you have to say anything, that you just like to add very briefly? We have probably a minute each. I'm not going to say anything further and then hand back to Tim. So I'm, after that little verbiage to get you thinking, I'm just going to go down as who's in the order on my screen. Simon, any last 1 minute thought?
No, not really, Tim. Just very quickly to say that some people would see any sort of control as a threat or somebody messing about with their breed. I don't see it that way at all. I think we work with science, we find out if there are problems, and we go from there. And I think going back to the points I think we've all made is communication, transparency, and using that science.
Tullis?
I think we've got to act now with our rare breeds. We can't wait. We just can't wait. You know, there's such good technologies out there. We've got to use them. Yes, I suppose we've got a slightly vested interest in it. And I saw some of the things. It's not just the male side and the sperm side. We can actually, even now with ovaries, we can slice them up and actually freeze them down to produce oocytes in the future. There is so much now we can do with tissue banking. And I think we've got to store it now. There's no point doing it in 10, 20, 30. Well, there is a point, but we need to be saving our breeds now. In 10, 20, 30, 40 years time, some of these breeds are going to disappear. So we have to use the science and technology. And some of these techniques, like the tissue bank, is very, very cheap. So it's acting now to save our fantastic and wonderful rare breeds for our future generations, really. Act now, otherwise they'll be disappearing.
Andy?
Thanks, Tim. What I'd quite like to do is just to pay recognition to the Horse Betting Levy Board and the support they have given over the years for our native breeds. Certainly from the Cleveland Bay point of view, it's enabled us to put in place not just the Sparks Breed Management Program, but to incentivize it and effectively provide the carrot rather than relying on legislation and a big stick. So, you know, there are organizations out there that are in a position to help support breeding programs. And like I say, particular thanks to the Horse Betting Levy Board for helping Cleveland Bays to get to where we are today.
And Paul?
So look, very much from a genomic technology standpoint of view, changing science, nearly unstoppable from an evolving perspective, and the amount of data that will come to the forefront over the next few years will be only ever increasing within the equine industry. It's about being able to leverage that data generation in a sensible manner, understand how it can be implemented into respective breed management strategies. But one thing for sure, it is coming. The technology is there. It's going to be here in the equine world very shortly, and that is a positive thing. More data will drive better educated management strategies, and the rest will be left into the hands of the breeders.
Great positive comment to finish on. Tim, if you could, I could hand over to you to draw this to a close.
Lovely. Thank you very much, Tim. I hope, ladies and gentlemen, you found the last couple of hours interesting, stimulating, and I think we should thank all the speakers and also Tim Morris for chairing it to bring all the various disparate bits together. So thank you very much, Tim, and thank you very much for the 4 speakers. I think it's been a really good session. But I think it's also worthwhile just recognizing these sort of webinars don't just happen without quite a lot of practice. So I'm very grateful to Vicky, Jan, Debbie, Beth, and particularly Georgina Crossman, the administrator for the forum, who've actually made it all happen. We get all the glory, or at least the public profile, but it's those good people who actually make it all happen. So many thanks indeed. And the same way, we couldn't do this without the support of the sponsors that we've had, in particular, the British Horse Society and the Horse Trust. So thank you very much. I hope you've enjoyed it. The next webinar is cunningly entitled The Great Weight Debate Equine. That's on the 27th January, Wednesday, 27th of January at 7 o'clock. You can find further details on the website. And the forum itself is on the 4th of March. And tickets will be available in early February. So if you do want to get involved in the actual forum itself, you can join our email list to receive the updates. So thank you very much, one and all. I hope you've enjoyed it. Have a safe, I was going to say safe journey home, but you're all sitting in your homes now. But have a good rest of the evening. Thank you very much.